In the blood of each human organism, there are antigens or agglutinogens that are embedded in the membranes of erythrocytes and antibodies or agglutinins that are in the plasma. Blood groups are an immunological characteristic that allows to distinguish people by the similarity of their blood agglutinogens. Currently, 36 blood group systems are known, such as AB0, Lateran, HEL, Rhesus, Diffie, KID, and others. The AB0 system is the first erythrocyte system of antigens that makes up the triallelic antigen system. This system is the most well-known and used to designate blood groups. According to this system, erythrocytes can contain on their surface two different antigens, A and B. Erythrocytes containing only antigen A are called erythrocytes of the second group, or A group, only B the third group, or B group. If the erythrocytes contain both A and B antigens, it belongs to the first, or AB group. If none of these antigens is detected, therefore the erythrocytes belong to the first, or zero group. It should be noted that the H antigen is present on erythrocytes, which is indicated by a capital letter. This antigen is always found on the surface of cell membranes in people with blood group 0 and is also present as a hidden determinant on the cells of people with blood groups AB and AB. A rare H gene is known, which is indicated by a small letter. When two H genes are inherited, the Bombay phenotype is formed. People with the Bombay phenotype do not have the H antigen, which is indicated by a capital letter, and therefore, in the presence of normal A and B genes, the production of A and B antigens is impossible. The AB0 and Rhesus blood grouping system is based on the principle of agglutination. Agglutination is the clumping of erythrocytes which is caused by antibodies reacting with the antigens of several erythrocytes and leads to the formation of the lattice that links the erythrocytes together. Agglutination of erythrocytes is observed if they are agglutinogens of the same name and agglutinins. In the blood serum, there are two different types of antibodies or agglutinins, anti-A or A and anti-B or B. Thus, anti-A and anti-B antibodies reacting with antigens A and B respectively will cause their agglutination. There are two methods for determining the blood group according to the AB0 system. The first method is to determine the group by the antigens contained on the studied erythrocytes. In this case, standard other gamma agglutinating serums or teleclones are used for the study. The second method is based on the determining of a group of erythrocytes antigens and serum antibodies. In such a study, standard isogamma-agglutinating serums, teleclones, as well as standard erythrocytes of blood groups A, B, and 0. Currently, teleclones are widely used to determine the AB0 blood group and Rhesus blood types. The use of teleclones provides reliable typing of the blood of the donor and recipient, regardless of the subgroup, strong or weak antigenicity of erythrocytes. In addition, the advantages of using teleclones are their safety, availability, cost effectiveness, and convenience. Teleclones are monoclonal anti-A and anti-B antibodies that are produced by two mouse hybrids and belong to class M immunoglobulins. Teleclone anti-AB is the mixture of monoclonal anti-A and anti-B antibodies. There are also anti-D and anti-C teleclones designed to determine Rhesus blood types. Scarify needle, anti-A, anti-B, anti-AB, anti-D, and anti-C clones, tablet, 0.9% sodium chloride solution, alcohol solution, cotton swabs, sticks. Mark the sections on the tablet indicating the name of the person and the specificity of the reagent. Apply anti-A, anti-B, anti-AB, anti-D, and anti-C clones on the tablet drop by drop, each drop containing about 0.1 milliliter. Take the blood from the patient's finger. Next to each reagent, apply one small drop of blood, about 0.03 milliliter. Separate clean sticks. Mix each drop of blood with the appropriate reagent to prevent false agglutination. Gently shake the tablet. Even in the first seconds we can observe a clear agglutination, but the result of the reaction should be taken into account 3 minutes after the end of mixing, so as not to miss weak variants of antigens. After 3 minutes, it is necessary to add 1 drop of saline sodium chloride to the drops where agglutination has occurred, in order to exclude non-specific agglutination of the studied erythrocytes. Write down the results immediately after the determination. With a positive agglutination reaction in the mixture, we see small agglutinates consisting of erythrocytes connected by antibodies. Small agglutinates gradually merge into large ones. In case of a negative reaction, the mixture remains uniformly colored red without presence of agglutinates. The examined patient's blood type 2 is determined according to the AB0 system because agglutination with clones anti-A and anti-AB is observed. According to the Rh system, a positive group is determined because there is agglutination with clones anti-C and anti-D. Fill out the workbook. You need to draw the results and write the conclusions.